Very nice introduction. Thank you. And I wanna go through a lot of little subject that we were trying to do our best to contribute, and especially to highlight what created an environment where we got so lucky to be able to make those contributions. So I just want to very quickly restate some of the points that, and I was changing my slides as we were going this morning to highlight some of the points that were mentioned, but that are tremendously important. And on this slide, what you see is the importance of infectious disease as a burden to public health compared to other problems. And it is a significant part of this entire pie that often we have a tendency to forget. And this is why I think this event here with Peter Piot being recognized is to tell you how important those recognitions are and all that work is, because it is the contribution that impacted on a lot of aspect of people's life. And this slide, it was to show you that there is 5 species of Ebola virus, and we got somehow unlucky because it's the Zaire strain that emerged in West Africa, which is linked historically to the highest mortality. But at the same time, we got also lucky because the Zaire strain is the one for which we had the highest amount of data and knowledge in terms of pathogenicity, but also in terms of vaccine development and even treatment. And if it would have been another virus, say Ebola, Bundibugyo, or Marburg, the level of advancement of those vaccines is definitely not as good as Zaire. So it was, in that respect, for our side, a bit of a relief that it was Zaire, although we knew that we were going to be up to the most aggressive of the Ebola virus. And I wanted to show this slide here because on this side, you see an infected cells producing Ebola. But here's a close-up, and it's actually a reconstruction with the computer. And basically the red molecules on top that covers the entire virus. So it's a virus that is like a filament, and from the name, filovirus. And what's red here that looks like little flowers is actually the proteins that are coating the entire virus, and that is what usually is targeted by most vaccine, and also an important part of the treatment, including the antibody-based treatment. And the reason is that by blocking the ability of or the function of that coat protein, it prevents the virus from replicating efficiently or replicating at all. And I thought this morning also what was an important fact is that we know a fair amount of what's happening here, transmission between human to humans, but there is a lot of questions here, and even the bat as a reservoir has been questioned. There is and which animal is amplifying. So there's a lot of work to be done here to really understand. And because most of the infectious disease are actually zoonotic, this has become more of a priority for the scientific community. So this is another way to see the outbreak in Sierra Leone and Guinea and Liberia, and it started here in Gekadu, as it was mentioned, in Guinea, and then spread to Sierra Leone and Liberia, and of course all throughout the Guinea, and this is reminiscent of a war zone, basically, where every one of those dots is an active chain of a center where there is transmission. And this is way before that, actually. This was dated October 30th. Before that, each one of those dots normally would require 40 to 60, at the minimum, expert to help controlling this outbreak. And there is no way that there was enough people to control all of these centers. And this is why in June MSF declared the situation out of control, because there was not enough. There was too many of those centers for the number of people. And this is the overall picture of the region. So this time the virus not only affected a small region, but the entire region, englobing 3 countries and passing 28,000 cases and 11,000 dead. So interestingly, we were trying to understand, as many others, retrospectively, what made this such a big challenge. And I think everybody will agree it's a combination of a lot of factors that made this a huge challenge. Interestingly, transportation within the 3 countries was relatively easy compared to DRC, and that also somehow promoted the spread. So because people were able to travel easily. In DRC, the locations are extremely isolated. They are often places where natural barriers like forests or rivers are basically trapping and restricting movement. And here's just pictures of river that we crossed with the lab early at the end of June 2014 after starting in Guinea and crossing to Sierra Leone to work with MSF and provide diagnostic. And this is just a very short summary of some of the challenges. And yes, communication with the population was a challenge. And yes, travelers also were a problem. But there was also a lack of experienced people. And this, I think the responders like me needs to also recognize where we can identify where we can do better. And on these pictures, this was all from the same location. And there was all mistakes that were fixed, but problems, for example, this ambulance driver had died because the PPE were being reused every day. And so this driver was helping the sick patients to get in the ambulance and then recycling the PPE and then finally got ill and died. This is another individual that was taking out the trash from the isolation ward being dressed in civilian clothing and just with a pair of gloves. This one receiving blood tubes outside, recording the information on the blood tubes and talking on his cell phone, despite the 2 pair of gloves. So again, these are examples, or this gentleman walking out of the isolation area and walking by with a contaminated mattress close to a person from the community, a woman. So again, this was adding to the challenge. So it's not only to say, well, it's because they didn't understand fast enough or we didn't communicate it fast enough. There was a lot of challenges that made this complicated. So we got a tremendous amount of support from the Public Health Agency of Canada to deploy. At one point, we had 3 labs in Sierra Leone, Kailaun, Magbaraka, and Freetown. And a lot of people from Winnipeg, from NML, not only from our section, but from other sections, volunteered to go to Africa to spend time and do all the work. And then again, the mobile lab concept was something that had been strongly supported by Frank Plummer, who is here in the room, as much as the vaccine. I'm going to talk a bit more about this. But what was interesting here is that as we were doing diagnostic and collecting a lot of data, we also were fortunate enough to go help in DRC at the outbreak that nobody has heard because it was at the same time and much more small and much more contained. And this brought the question, there are Zaire isolates viruses, and there is problem because of transportation, because of this, because of that. Are the viruses really the same? And here what we did is we compare people that presented at the treatment center with the same number of days, so there's no statistical significance. But when you look at these people at cases and the level of virus they have in their blood, they have more virus if they were infected by the virus from Sierra Leone than from the same, well, it's a different Zaire, but from the same species of Ebola virus in DRC. And so we thought, well, if people have higher level of viremia in their blood, are they releasing more virus into the environment? And Jim Strong, who is an active clinician in the group, started doing studies in treatment center in Sierra Leone. And with the help of MSF and coordinating with them, he started doing swabs and looking at where the virus was. And basically all those red dots is where the virus was found, including on healthcare worker, obviously. And that was highlighting the importance of removing the protective equipment the right way with the right sequence to make sure that there was no exposure. So we studied this also in animal models, and looking at one strain, what we saw is actually the strain from West Africa was growing at higher level, not only as we had seen in humans, but in animals in very controlled condition. And that again supported the concept that maybe that strain could be released in the environment a bit more efficiently. And what's nice is we got together with the level 4 community, and Heinz did another isolate, and Tom Geisberg did another isolate. And what's coming out of this is that some of the isolate like us are indeed growing at higher numbers. But some, like the one that Heinz tested, is actually a bit delayed. It's not as aggressive as the strain is comparing to. And I think that's very normal of a heterogeneous virus population, which we've seen with influenza also and a lot of other viruses, that depending on what isolate you look at. But so that being said, there is the phenomenon that some of the isolate are found in very high level in some cases. And we looked also over the time, so looking at July compared to November, at the same site. And these people also had presented at about the same days after onset of symptoms. And what we saw is an overall decrease. And when you plot it over time, what you see is that red bar, although it's in two phases, the people in November had overall lower level of virus than at the beginning. And what we saw at the same time, which is going to be published very soon, it's in press right now, is that actually in a population that is not representing the overall population, but in the population, and I'm going to tell you exactly what it is people that presented at the treatment center that were negative for Ebola. So they did have symptoms, but they were not infected with Ebola. In that population, the amount of antibody increased over time. And what we saw in December is that the cases of Ebola completely dropped. From 250 cases a week, we dropped to like less than 20. And we think that this may be one of the number of factors that may have contributed to this very rapid drop, in addition to good communication and good awareness to the population and all this. So we also were lucky enough to have enough support to not only focus on our main mandate, which is diagnostic, but also on doing research. And that, again, was in big part because of Frank Plummer, that gave us all the freedom and the ability to do that work. And in terms of therapeutic options against Ebola, there was a fair amount of work that had been done, but only two really had been tested in larger animal models. I'm talking about non-human primates when we started the outbreak. And this, for the antibody that was made, a treatment that was made in Winnipeg, it led to what was called initially the secret serum. And there was nothing secret about it. It's just that we were too busy with the outbreak to be able to publish the data. But actually, the initiation of this paper was actually in Sierra Leone. But again, and I wanted to show you a little bit more because I often have a question about the treatment. And what it is really is three antibodies that are acting like missiles and are attacking that protein on the surface of the virus and at three different locations, making the virus in a bad situation. It's very hard for the virus to replicate. And that's the goal, is to keep the virus low so that the body and the host can fight back and win that war. This was also, there was a lot of firsts in this outbreak. It's the first outbreak of Ebola where people were evacuated and infected, people that are coming from other countries. And as you've seen in the media, and this is highlighted here, a lot of people that were working and helping in West Africa were evacuated to different countries in Europe and in the US. And a lot of them received experimental treatment and or vaccine and also intensive care. So it's a bit, of a melting pot. It's very anecdotal, but I think sometimes from anecdotes, there's message to be retained. And this is a patient in Europe that received one experimental molecule here and then two injection of convalescent plasma and treatment, an injection at day 6 of the antibody cocktail, the ZMapp, and then the second one at day 9. And this curve was exactly reminiscent. These are level of virus in the blood of what we had seen in animal here in black, what you can see. And not only that, when you look at natural recovery and the number of days that it takes, these are all patients that receive it in a context of compassionate use, and you see that the number of days was shortened. And this was all before data and observation before the trial started. And overall, 24 patients that had received the treatment had tolerated the treatment quite well. And I think that the overall message I want to tell you, and I like this cartoon that I took from Dr. Teresa Tam, who is now the ADN, the Assistant Deputy Minister for the Public Health Agency of Canada. And this animal says, I'm going home today, they cure me using this new miracle drug. And I'm afraid it will be years before it's approved for humans. And this whole adventure, with, especially with the treatment, highlighted, I think, how good processes we have in terms of developing new vaccine and treatment in times of peace, when there's no emergencies, but how we need to have discussion how we can improve our processes to do the same job, but in times of crisis, and when the risk-benefit ratio is maybe different. And this is, I think, you know, what I want to highlight with this, and the next talk will show this even more, is that the regulators, FDA and Health Canada, that helped us a lot, did a lot of work, important work, and changed their way to work to make this faster. And it's amazing how much faster it did go, you know, from 10 years to maybe a year and a half. But let me put this in perspective. The 11,000 people that died in West Africa would have been the equivalent of over 100,000 people in North America, and another over 100,000 people in Europe. I don't know that we would have found a year and a half a fast-moving process. So I think that, you know, in the interest of me, who always think there's room for improvement, this is, I think this is something that could be part of future discussion. But the title of the talk is about how can we get better information from clinical data to inform on clinical trials. And this had been reported before, and what you see here is in red is people that succumbed from the Ebola infection, and in green, people that survived. And this had been reported for another outbreak, but it was very strongly associated in this outbreak with the level of virus in the blood. So people that reported to the treatment center with very high virus in the blood had very high chance of dying, basically. And so when you do a design for clinical trials, you can say, well, these people have close to 90% chance of dying. You know, do we want to have the same randomized trials than if we would deal with this group? And so these data, I think, in the future, hopefully will be also being used for this. And why I'm showing this, I thought this paper was fantastic. So this is once a year, the British Medical Journal, they have this one paper that is on the humoristic side. And so the title is indicating that basically what they were looking at is a randomized study of people jumping off a plane with or without a parachute. And of course it was not true, but it puts things in perspective that if you are jumping off a plane with or without a parachute, do you need to make this a randomized study or not? And by the way, jumping off a plane is not necessarily a death sentence. Some people have survived, not maybe voluntarily jumping off, but the parachute that doesn't open, although it is associated with a high level of mortality and side effect or long-term side effect. So I think, again, this has to be part of a future discussion of how these clinical trials are being designed and are being moved forward in a way that is also ethical. And very briefly, I'm going to show you a few things on vaccine, and I'm not going to show you much, but I wanted to highlight a few things. Is despite the fact that Ebola was not really a major public health problem before this outbreak in West Africa, there was a tremendous amount of work that was put into it. And thank you to the many governments that had put this on their priority list, and the many Department of Defense that funded that work. And you can see here, this is just a number. All these vaccines worked very efficiently in all sorts of animal models. Many which reach clinical trials, which is amazing when you think about it. Again, Ebola is not a major public health problem before this outbreak. And still, if you put it in side by side to HIV, to malaria, it is there's a bigger problem. We were lucky enough in Winnipeg. This was initiated by Heinz Fellman and managed after that by Stephen Jones. And Heinz's name is not there because I stole the slide from him. Well, he gave it to me. And you see some of the people that have contributed, but Stephen Jones has managed that project for a long time. Uta has contributed a lot as well. And others. Tom, we forget to mention him, but he did most of the work actually in the lab. And what it does is that it produces this bullet-shaped animal pathogen, which is a virus, can replicate, but instead of the normal virus, there's a piece of it, which is the envelope or the surface protein that is replaced, and in this case, can be replaced with the Ebola, and then it mimics. So the immune system sees that little part and reacts to it, and then you obtain protection against Ebola. And this can be adapted to Ebola, Marburg. We even have construct where you can put 4 antigen in the same vaccine. And I think here the vaccine field was, had the randomized trials on the table, but I think this was fantastic to see the ring vaccination strategy being put in the field, and as a matter of fact, is what paid off and where convincing data could be obtained in terms of potency. And I think, again, this was a good example of designing a trials in a way that was adequate to the environment and to get the maximum of information. And we've seen that slide is it's the first publication. I think we've seen that publication many times, the first publication that really shown in humans that the vaccine against Ebola can be efficient. And it's not that there is no, again, room for improvement. There is side effect. This can be further attenuated. The length of the protection is not known. And, for the emergency vaccine, it may not be that critical. But still, it's important to know, especially for lab worker like me that go in the lab, that go on outbreak, and where you want to have long-term protection. And it is a livestock pathogen. All the studies that have been done have not shown any problem on that side, but it's still being revisited just to be sure. And this is really, every time, I want to make this a tribute to me, the heroes of this outbreak, and they are the healthcare workers, especially the locals that I've seen their friends and colleagues dying, and they kept working, and the virus going through their own families, and they kept working. So I think that is very important to recognize. And not only the healthcare worker, but there was people like Jose here that is a WHO staff that trained countless number of burial teams. And we have a tendency to forget about them, but they also made a huge difference. This is a code, and I wanted to, now I can unreveal the code. Peter is not here, but I want to say that this little girl also had a coded message to say thank you, Peter. And on top of thank you signs. So I wanted to thank you for your attention.